Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Thank you for opportunity for us to sharing research information with all participant conference. Before I start presentation, I want to introduce my team in this research. Myself is Zulfahmi. I am from Agrotechnology Department, Faculty of Agriculture and Animal Science, State Islamic University of Sultan Syed Qasim Riau, Indonesia. Now I am a student, PhD student in 11 Maret University. Follow Mr. Parjanto, Edi Purwanto, and Ahmad Yunus. They are from Faculty of Agriculture, 11 Maret University, Indonesia. The title of our presentation is The Morphology and Density of Leaf Trichome Pasak Bumi, Eurycoma Longifolia jack in several natural populations in Indonesia. This is the outline of my presentation. Introduction. Eurycoma longifolia is a member of Cimarobaceae family. The species is widely distributed in Southeast Asian countries. In Indonesia, they are only found in Sumatra and Borneo Island. Pasak Bumi is one of the medicinal plants that have economically potential to develop in Indonesia, because the species have many secondary metabolite compounds that can be used as anti-inflammatory, antioxidant, antibacterial, anti-cancer, as well as increase male vitality. Trichome are uni or multicell which originate from epidermal cell of the aerial organ. There are significant leaf variation in morphological character, location, ability to secrete, and type of secretion. Currently, many researchers use trichome as a tool for taxonomic identification of plants, not only in generic classification level, but also in species or subspecies level. This is closely related to trichome character that are relatively stable, diverse, easy in preparation for study. Information about trichome morphology and density in Eurycoma longifolia species and comparison its variation between population in different environmental conditions is also not available. Therefore, we want to conduct the study on trichome morphology and density longifolia in the large-scale geographic. The objective of this study was to determine the trichome morphology and density in leaf of Eurycoma longifolia and its variation among populations, to observe its relationship with geographic and climatic factors of population, and to identify trichome characters that can distinguish among population studies. Material and method. Eurycoma longifolia samples were collected from 6 populations, 1 population from West Borneo, 2 populations from Riau Island, and 3 populations from Riau Province. 5 mature plants are taken from each population, which are selected randomly, and minimum distance among plants are 20 meters. Each plant was taken one leaf expanded fully and half. After collected, the leaves were then herbarized and sent to laboratory. For trichome collection, we use the Nile foliage method. After collection, trichome is observed using light microscope Nikon Eclipse 50i with 400 magnification. The image was captured on abaxial and adaxial lip surface using camera Nikon DS File 1 and analyzed picture using NINIS element software. For measurement, a total of the 30 images of each surface per population were captured. The general trichome terminology follows Sajna and Sunaj Kumar 2018 and Kaya et al. 2003. Trichome density was calculated by dividing the number of trichome with the view field area, according to Gonzalez et al. 2008. Analysis of variant, correlation analysis, principal component analysis, and clustering of the population were performed using SAS and Antisys software. Result and discussion. Based on our observation, we detected two types of granular trichome on abaxial and adaxial of lip surface of Longifolia, namely peltate and capitate. Both trichome are different in size, structure, and modes of secretion. Peltate trichome was composed of the one cell at the base, a very short stalk cell, and a large secretory head, like as a figure 1, and capitate trichome in the figure 2 until 4, composed by one cell in the basal, a star of the variable in length and uni or bicellular head, in which the head size is smaller than the peltate trichome. In this study, capitate trichome can be distinguished into three types based on morphology and dimension of the stalk, namely capitate trichome type 1, type 2, and type 3. Variation and trichome morphology and density between population. The result of analysis of variants showed that the number of each trichome and density were significantly different between populations in abaxial and adaxial lip surface, except for peltate trichome in adaxial. The difference in the number of trichome density between populations is caused by difference of environmental condition of population and genetic difference between individuals within population. More interesting, the density on the abaxial surface was higher than abaxial lip surface, 6.6 times higher than abaxial lip surface. It is maybe a strategy of land to deal with high light intensity attacking the plant lip surface, increasing sunlight reflection, and reduce water loss in the lip surface. The coefficient correlation analysis between trichome morphology and environmental factor. Based on the table, we can see all the coefficient correlation value between trichome morphology and density with side environmental factor were positive on and negative correlation, depend on trichome type and surface position. More interesting, the result of the correlation analysis confirmed that high trichome density in adaxial lip surface is closely related to temperature. Adaxial lip surface receive higher temperature than abaxial lip surface, and plant will present higher trichome density as a defense mechanism against excessive radiation and higher temperature. Martinez Natran et al. 2018 explained that the secretion of chemical compound from trichome become a protector of plant photosynthetic tissue. The secretion reflects excess solar radiation and help absorb the heat. Imply lab temperature are maintained near optimal level for photosynthesis and carbon fixation. To know the contribution each variable in separating a population, the principal component analysis was conducted. Two principal PC1 and PC2 explain 90.8% of the total variation among population. PC1 explained 91% of the total variation, in which the highest contribution character was trichome density in adaxial lab surface, while PC2 explained 8.8% with the highest variable contribution was also trichome density in abaxial. This result indicates that the variable of trichome density can be used for separating a population of the species studies. The scatter plot of the PC1 and PC2 from PCA analysis can separate population of Eurycoma longifolia into four clusters. The cluster 1, Sentaja population, cluster 2, Tahura population, cluster 3, Lingga and Fokomo population, and cluster 4, West Borneo and Lingga 1 population. The dendrogram UVGMA confirmed the projection of the scatter plot from PCA. In which the coefficient similarity among population was 0.14, UPGMA dendrogram divided population into four groups. Conclusion: two types of granular trichome were detected in leaflet Eurycoma longifolia, namely pelted and capitate trichome, and there are significant differences among populations. Trichome morphology and density have a positive and negative correlation value to climate and geographic factor of population. The variable of trichome density can be used for a separating population of study. Acknowledgement. The author would like to thank the LPDP Ministry of Finance Republic of Indonesia for funding this research, and Forestry Service in some region for permission of the collecting material in the field. Thank you for attention all participants. Wassalamualaikum warahmatullahi wabarakatuh. I back again to committee.